Testing. Okay, can the last person hear me? Okay. All right, good afternoon, everyone. Thanks for having me here. I'm very grateful for this opportunity. Today, I'll be sharing on our team's work on predicting time to gastrostomy in ALS patients. So I have no financial disclosures. Currently, I'm working and training as a rheumatology senior resident at the National University Health System in Singapore. And I started working on this project with the team earlier this year during my internship with the then SICS, now Singapore Institute for Human Development and Potential at A*STAR in Professor Dennis Wang's lab. I would like to start by giving an overview of the disease ALS and then move on to talk about the data used, as well as the models for predicting the gastrostomy event. I will then demonstrate how the tool is used, as well as discuss further work. So ALS, which is amyotrophic lateral sclerosis, is also known as motor neuron disease. It is caused by the degeneration of upper and lower motor neurons, and its prevalence is around 6 to 9 per 100,000 persons. However, as the prognosis is generally poor, with median survival of ALS patients between 20 to 48 months, the lifetime risk of this disease is around 1 in 350. And it can present in different ways. Around 1/3 of patients have bulbar onset disease, around 5% with respiratory disease, and just under 2/3 with limb onset disease. Management of ALS is through a multidisciplinary approach targeting patients' symptoms. The ALS Functional Rating Scale, which is the ALS FRS-R score, is used to assess function of patients, and a reduction in a patient's score indicates a decline in physical function. There are 12 items in 4 domains on the right, with a maximum score of 48, where each item is scored from 4 to 0. From the literature, we know that these are the various negative prognostic factors for survival, which include malnutrition, weight loss, later age at onset, and bulbar onset. When a patient's swallowing function declines, they are unable to take food orally safely. So initial management can include dietary modification of food textures, industrialized thicker fluids, as well as rehabilitation. And evidence shows that earlier gastrostomy tube placement may prevent further weight loss and improve survival. Time from symptom onset to tube insertion ranges widely, from 13 to 24 months in one study to an average of 46.9 months in another. And the reason for this is because that multiple factors influence the decision for gastrostomy tube insertion. So while treating ALS patients, there is a need to balance progressive nutritional decline, resulting in patients progressive nutritional decline requiring tube insertion against the risks of tube insertion. So we set out to ask the following questions. Number 1, are there distinct trends in patient trajectories of decline? And number 2, can we predict for a patient what is the likely time of gastrostomy tube insertion? And ultimately, we hope to better plan for patient care. The data was obtained from 3 different databases: PROACT, ARQ, as well as IDPP. PROACT is the Pooled Resource Open Access ALS Clinical Trials, the largest publicly available repository of merged ALS clinical trials, with data from 29 phase 2-3 clinical trials. And ARQ is a local database of ALS patients where our team in Sheffield have collected data longitudinally. The third database is IDPP, which is a curated dataset used for the Intelligent Disease Progression Prediction Challenge dataset, which consists of data from 2 clinical institutions, Lisbon in Portugal and Turin in Italy. Baseline demographics for the patients were collected, and clinic measurements were collected over time in terms of weight, respiratory function, total ALS-FRS score, as well as subscores of bulbar and Q3, which represents swallowing function. Together, we have data from over 8,000 patients included. The mean age at onset was 57.4 years, and 67% had spinal or limb onset. Around 1/4 of patients had gastrostomy tube inserted. And so the first question was, were there any distinct patterns of decline in the patients? We focused retrospectively on part of the data with at least 3 time points after the onset, and used the unsupervised classification method to understand the patterns of decline of total ALS-FRS scores and subscores of bulbar, Q3, as well as respiratory marker in ALS patients. The next step was feature extraction, where we looked at the classes and exponential decay. We then proceed to prospectively predict the time of gastrostomy event for each individual patient using random forest and XGBoost predictive models. Finally, we incorporate the elements of these steps into a visualization tool to predict individual patient trajectories. Now, firstly, to understand the patterns of decline in the patients, we use the unsupervised classification method of joint growth mixture modeling, discrete time survival analysis. And this reviewed distinct trajectories of decline with corresponding probabilities of gastrostomy insertion. There were 4 distinct classes in total for the ALS-FRS as well as the bulbar score, the very rapid, medium, and slow rate of decline. And in the respiratory marker as well as Q3 score, we saw 3 different distinct classes. And for the weights, we observed that there were patients with rather stable weights versus patients with declining weights. And we calculated the slope parameter for each time point and assigned binary numbers, 1 for patients with at least 5% weight loss from baseline, and 0 for those that did not reach 5% weight loss from baseline at that specific time point. And we then move on to prospective predictions of individual patients. We sought to predict the estimated time of gastrostomy within plus minus 90 days of the actual event, using random forest as well as XGBoost models. All the longitudinal parameters, such as ALS-FRS score, subscores of bulbar, Q3, respiration, and static parameters like age, sex, diagnostic delay, site of onset were considered, where over 200 parameters were used to build the decision trees. And for the random forest, the accuracy was 71%, and for XGBoost, it was 72%. This is a working model of our visualization tool that we are developing and constantly improving. On the right, there is an input panel where the clinician can include the patient's age, sex, weight, ALS-FRS scores, as well as other measured variables. This can be submitted directly through the tool or through uploading a CSV file, and these can be included for various time points for the patients. Thereafter, the tool shows the patient's expected trajectories as well as the predicted time to gastrostomy. I've just started the video, so it should submit the data. The tool is work in progress, and we plan to update it with our latest predictive models. So I'll move on to talk about the strengths and the limitations of our study. The strengths are that we have included both clinical trial cohort data as well as hospital data. And we have endeavored to make a prospective prediction of a gastrostomy tube insertion within 90 days of the actual event. Some limitations of our data is that as there was incomplete data across the various time points, what we tried to do and to overcome this was that we used the data that had more time points for assessing trajectories at the very start. Other aspects of management that we did not include were whether there was any use of disease-modifying drugs. Now, some further work that we plan to do is in terms of updating our tool with further analysis, and then we also want to have some conversations with patients and healthcare providers as to how the tool can be better used to plan care. In terms of working on other aspects of ALS, we could also use it to predict other outcomes in ALS, such as time to initiation of NIV, non-invasive ventilation, tracheostomy, or death. And in terms of use in other diseases with declining trajectories, such as neurodegenerative diseases such as dementia. Okay, so these are my references. And huge thanks to our team, Chris, Dennis, Harry, Juan, and Varsha, who is here today, as well as the rest of the lab at then SICS, now IHDP. So some of them are here present as well as the rheumatology division at my hospital for supporting my work. Thank you. Yes, so we split 70-30. Yeah. Looks like we don't have any more questions, so thank you, Jane. Thank you.
So our next presenter is Dr. Yurui Chen.
Thank you. Hi, everyone. Thank you. So I'm Yurui. I'm from the National University of Singapore. I'm at the Maths department. I'm a final year PhD student there. And right now, I'm also working at the Institute of the Human Development and Potential, A*STAR. So today, I'm going to present my research titled Predicting Early Life BMI Trajectories through Proteomics Data with Extended Dynamic Mode Decomposition using deep neural networks. So the influence of the obesity in early life can persist in like the adulthood and contribute to an increased risk of chronic diseases in later life. The rising prevalence of child and adolescent obesity poses significant challenges to optimal the human development and health longevities. So from the previous study in Singapore, 70% of the children in Singapore who are overweight at age 7, they stayed overweight after they grown up to the adults. So it's quite important that the prediction of the body mass index trajectory in the early life is essential for the early management of the obesity. Well, much of the existing research and the models has concentrated on the analyze to the find some the proteomics biomarkers to the BMI itself. It's yet the dynamics between the proteomics dynamics and the progression of the BMI have yet to be fully explored. So in this research, we would like to build a machine learning models to first modeling the BMI trajectory of the babies, and also using the omics data for the final prediction of using the historical trajectory of the children from year 3 to year 8, and finally we can predict the BMI at their year 9. We adapted the data from the cohort of the growing up in Singapore towards healthy outcomes GASO study. Right now we have 355 children, where half of them are girls and another half are boys. So we selected subgroups is because they have the complete BMI measurements from year 3 to year 8. And also they got a proteomics measurement at their year 8. And this proteomics is coming from the 92 neurology related proteins, which were measured from the O-link in their year 8. So our final goal of the model is the prediction of the BMI at their year 9. We use the historical data and the proteomics trying to reconstruct their BMI trajectory and also finding the future trajectories. So how do we solve this problem? We'll start from the data-driven dynamics predictions with a mass formulation. So mathematically speaking, let X be the state space and U be the parametric space. So this X in our problem is the proteomics trajectory, and U be this X being the BMI trajectory and U being the proteomics. So what we want to is to find a continuous function f which transfer the state space and times U is a control space. F maps the X times U to the space X. And from the observational data, what we have is here, for the sample J, and they have different time points n, and with its own proteomics, and we would like to find a function f that using the transformation of x to using the historical BMI, we can get their new BMI xn plus 1 for this sample J. So our goal is if we can have the initial stated of the BMI at their very early ages, we can predict their grown up BMI. But here, one of the challenging things is f might be nonlinear, and right now we only have the separated discrete observation of the x and u, but it's hard to map them to a continuous function. So the idea is we're trying to map the this nonlinear continuous function to infinitely but linear spaces. So the method we are using here is called extended dynamic mode decomposition using the Koopman operator. With the same set of input data, we first build a dictionary function. It's called psi. So it's a basis span. So this space of psi is a span of the basic function of the dictionaries. And using this nonlinear dictionary, we first map the BMI itself to a higher dimensional vector spaces. And in this higher dimensional vector spaces span psi, it has a linear dynamics. So if we consider the psi xn plus 1, it gets a linear dynamics relationship between the psi xn, which is modeled by the Koopman correlator k here. And how do we solve this k in our model? So we do the optimization and use the pseudo inverse or doing the parametric modeling. So with this assumption, and we can have after mapping the xn to the higher dimensional spaces, we have the linear dynamics, which is easier to solve. But here is still some challenges. First of all, it's it's harder to decide the dictionary spaces here. What kind of dictionary basis we have, to find optimal solutions. And another is this k might not be a constant, but might be depending on the u itself. So next we do one step more. We do the parametric Koopman operator learning. So we solve these questions by 2 steps. We build separate neural networks to parametric the dictionary psi and also the projected operator k here. The k, depending on the proteomic itself, we feeding them to a neural network, and also the psi, which maps the BMI trajectory to the higher dimensional spaces. We also use another neural network to represent. And our full model looks like this one. So the input for disconnected. Okay. So the input of the model is the historical BMI trajectory and also the proteomics data. The loss function is computed based on the difference of this one is mapped for the xn plus 1, and this one is a prediction for this xn plus 1, but using the parametric Koopman operator. So this loss function is optimized based on all the time step n and all the samples j. So afterwards, we can reconstruct this historical trajectory of the BMI, but also do the prediction of the new BMI in the future. So here are some model performances. We build a sex-specific model for the boys and girls in the GASO study. So right now, we can see that we have the RMSE of less than 2 and Pearson correlation at 0.85. And there's another message I want to convey is this dashed line is 85% of the BMI, meaning that if this x-axis is a true BMI at year 9, if the boys BMI at year 9 passes this threshold, is considered as overweight. So in our model, all the overweight boys is captured based on our models. So although it's not perfectly predicted by the regression, but still can capture the risk of overweight from the model. So the result is a bit better for the girls model. We can see all the overweight girls are captured here, and also the PCC is better than the boys. And we do the model comparison with 2 models. The first one is commonly used elastic net when doing the omics risk predictions. And another one is a neural network, which can model the nonlinear relationship between the proteomics and the BMI outcome at year 9. So in both of the settings, the boys and girls, our proposed model, EDMD proteomics, outperforms the other tools. And especially if we check the results in more details, these are the Go models using the elastic net and the neural network. We can see that the prediction is quite noisy and it cannot capture the overweight results here. Okay, that means that with the integration of the proteomics and the historical BMI, we can reconstruct the trajectory and make the fine prediction. And then we try to detect some novel biomarkers of the children, which is related to the childhood obesity. So we use a lime to do the feature explanation and found that these are the top 10 most important proteins here. And one of these, alpha-2 MRAP, is also found quite related to the obesity even in the adults, so in previous papers. And I want to emphasize that it's not only the prediction from the explanation, because it also comes from the reconstruction of the trajectory. So meaning that our detection of the biomarkers is not only depending on a simple regression, but also depending on the trajectory of the BMI in the childhood. Okay, so here are some limitations and the future works. First of all, recall that our model can use the dynamics control UN here, but we only treat it as a static value since, I'm sorry, what happened? Okay. But we only treat it as a static points here because we only have the measurement of the proteomics at year 8. But if we can has a longitudinal data of the omics profiles, we can make the models more reasonable. And there's a recently newly published paper and the open public available resources for the nonlinear dynamics of the multi-omics profiles. But it's only for the adults from the age 25 to 65. So it's also very important if we can get omics profiles in a longitudinal manner for the children for better model construct. And also, we need to do more benchmarking with not only the proteomics model, but also the trajectory-based model using only the BMI to see what happens. And also, we can do the biomarker detection by analyzing on the operation Koopman operation key here because there are some spectral information provided in this Koopman operator. So it's quite important. And finally, I want to say thanks to all my supervisors, mentors, and my colleagues here. So this is my supervisor, Prof. Zhang Louxing in the NUS Maths department. And also I want to thank Dennis, who is a PI of our team, and also Pan Hong, who is my mentor, and our colleagues in SICS, the same group of Jane. And also I want to say thanks to Prof. Qianxiao Li, who is in the math department, and Mrs. Guo Yue for the useful discussion of the Koopman Decomposition series. And if you have any questions, you can ask here. And also, if you want to find more interesting projects, I'm also interested in the AI drug discovery and some risk prediction models. So you can check my GitHub repo and contact by email. Okay, thank you so much. Hi.
If I understood correctly, you are predicting BMI at age of 9. Is that correct?
Yeah, so the model is evaluated based on this metric, but the model is actually constructed based on the reconstruction of the historical BMI and also the future trajectories.
Well, I'm not clear what's the clinical advantage of predicting BMI at age 9.
Oh, so here I want to showcase this model can be used for any phenotype trajectories prediction, but right now we have the BMI is a complete set. So if we have the continuous measurement of other diseases, even the diseases, we can see the trajectory of the disease onset. So BMI is just one example of the usage of this model since it's available in our data set. So right now I'm trying to integrate the historical phenotypes and also the trajectory of the omics. So that might be the clinical advantages. And yeah, does it make sense to you?
Yeah.
Yeah, thanks. Hi. Yep.
Hi, nice presentation. Thank you for sharing. I have looked at that book, so this is interesting. Personally, 2 questions. You mentioned earlier that your proteomics panel is from the neurodevelopmental or neurological
Yes.
proteomics.
Yes, true.
So maybe can you elaborate a little bit on that? And then the second question is how do you account for potential early, I lost the word, puberty, because the girls are more likely to have earlier puberty than boys. So what's the oldest age group of your data set that you can discuss? Thank you very much.
So for the first question, could you remind me the question, the first one? Oh yeah, these are I think it's a predefined neurology panels for these proteins. There are 92 neurology phenotype related proteins so in this panel, and we only measure the proteomics of this 92 proteins, not a whole proteomics panels. Because this data set is also related to the other studies, so I'm only using this proteomics panels here. Yeah, and for the second one, could you remember the second one? You mean the age range of the girls and boys?
Especially for the girls.
Sorry, Jian, could you help me with it?
Some girls may
Yeah.
have early puberty, and then their the trajectory will change quite a lot. So how do you account for that?
So I didn't specifically account that for the model, so just let the model learn itself. Yeah, so maybe I need to check these extreme cases. Right now I didn't. But right now from the model from the model performance, it can say that if like I didn't show the trajectory here, but yeah, I actually saw some like extremely changed trajectory. It can be captured by these nonlinear models. So it's already, this nonlinearity already been trained in the Koopman operator since it's a nonlinear model based on neural network. Yeah, I will check that. Thank you for the interesting questions. Yeah. Hello.
So that was a nice talk. I'm really interested when you were talking about using the neural net to model like the nonlinear relationship.
Yeah.
Are you interested in rederiving the actual mathematical relationship? Because I think that is important because BMI is driven by both height and weight.
Yeah.
So if it's just a neural net and it's a black box, we may not always be able to catch that.
Yeah, so it's actually not so neural network here is only our parametric model. So really mathematical model is called this EDMD model. So we can start from here. So it's a functional, it's a trajectory approximation actually. So we trying to find approximation of this nonlinear function f, which is the trajectory of the model. So what we do here is we use like the functional dictionary of this phi. We try to construct like a span of these functions, like m functions. And after we map the BMI to the higher dimensions, there will be like the linear dynamics in this higher dimensional system. So this is the math part, and it can be shown that if we select a very suitable dictionary, it can be achieved that we can like map this nonlinear f to this linearly trajectory in the higher dimensions. So what we are using for the neural network is not we directly learn the proteomics or the BMI. We use this neural network to represent here, to just parametrize the key and the dictionary functions. We can also set up dictionary functions, but this neural network is only like the approximation for this, for the automation of the selection of the basis function of the dictionaries. So yeah, so it's not like directly the black box, like just different models. The basic mathematical part is like we can map the nonlinear functional trajectory using a set of dictionary like by the high dimensional mapping, and it will like fall into the linear dynamics, and we can easily solve the linear dynamics. So that's the mathematical insights here.
Okay, so in this case, you can actually pull out the function.
Yeah, true.
And replot it.
Yeah, true. So right now here I show this size can be spanned by the neural network, but it can also be spanned by some like explicit functions if you want, like the RBF kernel here. We can use the RBF kernel of the BMI, like mapping them use the RBF kernel to like the higher dimensional spaces. Yeah, thank you.
Thank you so much. Any more questions? Okay, we're going to go to our next presentation. Thank you so much. Here we go. All right, is Dr. Konstanze Tan here? Are you presenting on his behalf? Okay, perfect. Or, oh, sorry. Come on. No worries. Yeah, that's what I have next. Let's see, yeah. Also, let's see. This is you,
Okay,
right?
yeah.
This is you? Okay, this is your presentation?
Okay.
Okay, perfect. Here you go.
I think, can I connect my clicker?
Sure.
I have a, yeah. it's not working, I can just.
Try, see? Oh, it's working.
Oh, okay. Okay, so good afternoon, everyone. I'm Konstanze from the Lik Hang Wu School of Medicine, and today I'm very excited to share my experience with using multi-omics data to unravel the pathogenesis of dilated cardiomyopathy. Okay, so I'm focusing on epigenomics and specifically DNA methylation, which can be influenced by genetic variation and also environmental factors. And this in turn leaves changes on the epigenome, which affects gene expression. So the specific mark that I'm studying is DNA methylation. And this is one of the more well-characterized epigenetic marks because it is stable and also amenable to high throughput profiling methods. And so this involves the addition of a methyl group on top of the 5' cytosine. So in DNA we have ATCG. So this is a modification on top of C, and it converts it into 5' methyl cytosine. So this epigenetic mark in particular has been studied in various disease contexts and also in cardiovascular disease. And dilated cardiomyopathy, which is characterized by a thinning and weakening of the left ventricular walls, is one of the etiologies of heart failure and is notoriously heterogeneous. So what this means is that even though there's no clear cause assigned for most of the cases, rendering it idiopathic, and even though more and more we are finding that the cases have a genetic basis, but this genetic basis still doesn't completely explain the various manifestations and severity of disease. So even for genotype positive patients, they can vary a lot in how the disease manifests. So this has led researchers to explore DNA methylation as a different avenue for probing the pathogenesis of dilated cardiomyopathy. So the history of this really started in around 2010 with a small single-digit sample size, and as the years progress, we are able to get more left ventricular tissue samples. And not just the increase in the number of sample sizes, but also the maturation of methylation array technology, which means that previously, we were only able to see these methylation alterations at regional resolution, so like peaks. But now we are able to get a single CpG resolution thanks to the Illumina arrays. And so since we had our hands on the largest series of left ventricular tissue available, as well as the newest array technology, we decided to expand the methylation signature of dilated cardiomyopathy. And methylation is not the only interest, because the reason why we're studying methylation is because we want to know more about the pathways that are regulated by methylation. And one thing that comes straight to mind is gene expression, because that's the most direct link that we know. So the studies have all been incorporating expression changes as well to understand how these methylation changes that are linked to disease, what genes might they regulate. So in recent years, we have also seen growth in the number of quantitative trait loci studies, which means the relationship between genetic variants and methylation. So this, plus the availability of new statistical methods for causal analysis, it enables us to take a step beyond association to see if these relationships between, for example, methylation and gene expression might be potentially causal. And I will explain more about this statistical technique for causal analysis in my next slide. In the slide after this. But for now, I will be introducing the cohorts that I've been using for my study. And so I have two independent dilated cardiomyopathy cohorts, and this allows me to not just discover, but also replicate the changes in different cohorts. So for my cases, I have biopsies from patients with dilated cardiomyopathy, and for controls, they are obtained post-mortem. So DNA and RNA are extracted from these samples and used for methylation profiling, genotyping, as well as RNA sequencing on the same samples, and this provides the multi-omics data that I have been using for my investigation. So the study is designed to answer three questions. Firstly, which DNA methylation sites are altered in dilated cardiomyopathy? Basically, which CpGs are linked to dilated cardiomyopathy? Which genes do they affect? And also, which of the relationships in 1 and 2, which are associational, might be potentially causal? So to answer these 3 questions, I was using multi-omics data from the 2 independent cohorts. And the first step really involves an epigenome-wide association study. So many might be familiar with genome-wide association study, but since I'm using CpG methylation, this is an epigenome-wide association study. I first performed a discovery stage analysis in one of my cohorts, and the top hits are then taken forward for targeted replication testing. And together, this identifies 194 DCM-linked CpGs that are independently associated with DCM and directionally confirmed in both cohorts. So to find out which genes they regulate, I conducted expression quantitative trait methylation study, which is essentially the association between CpG and gene expression. This was also done in the replication cohort and giving me a set of directionally replicated CpG expression relationships. And finally, since I have genotype data also available, I looked for genetic variants that were linked to the CpGs. And this could be used for Mendelian randomization analysis, which is the causal statistical technique that I was alluding to in the previous slide, and which I will now explain. So this is a technique that I'm using to take me one step beyond association to gain some just preliminary insight into whether this relationship might be causal. So this is Nature's randomized control trial, and as we are familiar with, randomized control trials are the gold standard for assessing a relationship between an exposure and outcome, and the exposure can be something like drug treatment. And the essence of the RCT basically is that it randomizes people into treatment and control groups depending on whether or not they take the drug, and the outcomes are then compared. But for my exposure of interest, which is DNA methylation, it is not possible to edit the methylation status of many genes in humans. And so, but actually nature has a natural randomized process for this. And this is why I introduced the concept of genetic variants that were linked to CpG methylation. So because of meiosis, an individual will randomly inherit the genetic variant that is linked to the CpG. So in a case, this is an inherent randomization process that's already present in nature. And this concept forms the basis of Mendelian randomization. So, but a specific kind of Mendelian randomization I'm using is a tool called summary data-based Mendelian randomization. So called because summary data, because it incorporates genetic association studies like GWAS studies, MEQTL studies to get the effect size estimate for a genetic variant on the disease and on the trait that I'm studying. So this method calculates a causal estimate, which is called beta SMR, for every single CpG site that I'm studying in relation to DCM or in relation to gene expression. So I'm doing 2 separate causal analyses, one for dilated cardiomyopathy, but one also for the genes, for specific genes that might be future experimental targets to probe DCM pathogenesis. So this is the first main finding of my study. Is that there are 194 independent signals, actually all of them are hypomethylated in DCM, and they are able to effectively stratify case and control. So you can see the 2 distinct classes that are found there. So this indicates that these 194 sentinels are capturing the main disease-linked methylation changes that segregate the 2 groups. And so I followed this up by causal analysis, which between the CpG and the DCM. So it suggested that 2 sentinel CpGs might have causal links to DCM. And I would like to draw your attention to CG08140459 because it was not just reported by us, but by previous publications to be associated with DCM. So this was, it has an inverse association with DCM, meaning that hypomethylation of this CpG increases the odds of DCM. But we also wanted to find out the target genes. So out of the 194 sentinel CpGs, we examined their relationship with cis genes, which are proximal genes within 1 megabase distance, and 183 of them had replicated relationships. And this also applied to the CpG, which I mentioned in the previous slide. So previously we found that it could have causal links with DCM, but now we find we take that causal relationship, we add some more granularity into it, and we find that it regulates LTBP2 expression. Specifically, this is inversely related, meaning that hypomethylation of the CpG is associated with increased LTBP2 expression. And LTBP2 has roles in myocardial fibrosis and DCM prognosis. So this just provides a flavor into the fact that our analysis is capturing some known aspects of DCM pathogenesis. But we didn't just identify known genes. We found some evidence for other genes which have not really been well studied in the cardiac context as well. And we found this via, so this is causal analysis of the relationship between CpG methylation and gene expression. And this led us to find 36 sentinel CpGs with possible causal links, which means a significant SMR estimate with 43 unique proximal genes. And we also pursued something called co-localization analysis to find out, to provide us with an inkling into whether a single causal variant might be underlying these this association. So that could be, the causal association that we see could be a result of another scenario whereby actually variant 1 links to trait 1 variant and another variant links to trait 2, but these two variants are in linkage. So it's not a single causal variant underlying both of them, but it's actually in linkage, which is not really of biological interest compared to the single causal variant scenario. So this co-localization analysis plus SMR analysis led us to, so I'm just showing the top hit, which has the highest posterior probability of co-localization between the MEQTL and EQTL signals, as you can see on the panel on the right there. And it also has significant SMR causal estimate for relationship with IER5 gene expression. So IER5, as far as we know, has not really been studied in a cardiac context, but is involved in DNA damage response and also notch signaling, which make it maybe a promising target that we want to look into if we are trying to design experimental studies. So beyond that, just now I mentioned that we use the methylation array to profile methylation sites, but this is only a very limited portion of the epigenome, about 2 to 3%. So we pursued targeted resequencing, which just means we want to capture more CpG sites that are around the CpG sites that are the limited CpG sites on the array. And we didn't do this in the DCM cohort because we only had data in another cohort. But we decided to just so this other cohort that I'm referring to is an iHealth T2D cohort. It's a general population-based cohort, not a DCM cohort. But it was very well phenotyped for a bunch of preclinical and clinical cardiovascular traits, for example, blood pressure or elevated systemic inflammation. And so with this increased number of CpG sites, we decided to see if we could identify any new signals around the DCM-linked CpGs that might be linked to any of these cardiovascular traits. And I'm showing an example here. So as you can see in the purple diamonds, these are the CpGs that are linked to DCM, but around them, like for example, the red, the top hit in this region, it is not on the epic array. But because of this targeted resequencing, we were able to capture this signal as being associated with preclinical cardiovascular traits. So the point of this slide is just that if we use methylation array for looking at disease traits, we might want to consider targeted resequencing around them to capture more signals that might be potentially relevant. And beyond that, I've been talking about single CpG site association, but we also perform modular analysis to identify if there are any coordinated changes in CpG methylation patterns. And using WGCNA, which is a rated gene correlation network analysis, we identified 6 modules that are co-methylated, and I examined each of them for gene set enrichment as well as enrichment in transcriptional factor binding sites. And so the pathogenesis of DCM can broadly be conceptualized as cardiomyocyte intrinsic, which happens within the main cells of the heart, or other cardiac cells like fibroblasts or immune cells. And what we found is that these certain modules were enriched in specific aspects of DCM pathogenesis, such as, for example, module 6 was enriched in genes that were involved in altered metabolism. And some of the modules were also enriched in the corresponding transcriptional factor binding sites that are known to have a role in the metabolic processes, for example, or extracellular matrix processes. So this provides an overview of the various omics analysis that were performed on the DCM Sentinel CBGs. And currently I'm working on using cardiac chromatin interaction data, for example, high-C data, to validate my expression quantitative trait methylation loci relationship. So let's say I've observed an relationship between CPG and gene expression, but is there actually physical evidence that these two regions interact in the cardiac tissue? And with publicly available data sets, I'm now able to look at this. And importantly, my analysis was conducted on cardiac tissue, which is heterogeneous, has many cell types. So I don't know which, I haven't done analysis yet to clarify which cell types are responsible for these mechanisms, but that's something I'll be looking forward to pursuing in future. And I would like to thank the participants and study participants, individuals and consortiums who have enabled and sustained this investigation. This presentation is based on a preprint publication that you can check out if you scan the QR code. Yes, thank you for your attention, and I'll be happy to take questions. Maybe I'll leave this slide. Yeah.
No, that was very clearly presented. Just a question. You mentioned that most of your CPGs were there was hypomethylation enriched.
Yes.
In DCM. Why is that? Because, I mean, is there any hypermethylation associated with gene downregulation? Does that mean that all your genes are upregulated in DCM, like overexpressed?
Thank you for that question. I will acknowledge first that my methylation array captures only a very limited size of CPGs. So I think if we have used methods like maybe whole genome bisulfite sequencing or something more comprehensive, we might be able to capture more CPGs that maybe exhibit different patterns of methylation, hypo or hypermethylation. But another reason I can think of is that this methylation array is gene-centric, meaning that they focus on promoters. Yeah, so typically these tend to be hypomethylated in order to increase gene expression. But methylation at other areas of the gene, like gene body, the relationship is not so clear, and it will be interesting to investigate. So thank you for your question. Yes, thank you.
Have you seen particular CPGs associated with subsets of DCM? I mean, for example, some that might be just present with, say, I don't know, Titan variants or something like that?
Oh, yes. No, I mean, okay, I haven't seen it, but I said yes because I found your question very interesting. But actually, the main, that would be an interesting angle to investigate, like to see if CPG methylation is involved in pathways, because we have done some GWAS, there have been some published GWAS for DCM, and we know that certain variants and certain genes are associated with DCM. So one angle to pursue is whether these genes might act on DCM through CPG methylation. That is not an angle that we have pursued in this. It will be interesting as a potential analysis. But for this investigation, our focus was on using an epigenomics perspective to find maybe genes that were not suggested by genetic studies to be involved in DCM. And in fact, from the timeline of epigenetic studies that have been conducted on DCM, most of the genes that they identified and also validated in vivo to be relevant for cardiac pathogenesis were not the same genes found by genetic association studies. So, but it will be definitely interesting to examine the intersection between the two. So thank you for your suggestion question. Yeah. Yes.
Great presentation. Quick question. You want to look at cis genes, right? CPG and the DNA
Yes.
genes. But what if there's a gene that regulates multiple CPGs in trans? Have you thought about doing CPG and trans gene expressions? So see if there's a master regulator of this DCM-related CPGs, and that's how that is progressing.
Oh, do you mean if the CPGs regulate genes in trans?
Or there's a gene that regulates multiple CPGs in trans?
Yeah, I think trans analysis will be something that I'll be interested in pursuing. It's just that for this preliminary analysis, since we know the relationships between cis CPGs and cis genes are clearer, we wanted to get a flavor of that first. But yeah, the master regulation is of interest as well. Yeah. And just to add, for the master regulation part, that might be an interesting expansion for my WGCNA analysis, because that looks at co-methylated CPGs, which may be more relevant to master regulatory networks than my current cis CPGs. Yeah, thank you for your question.
All right, everyone. Thank you so much. Thank you.
Thank you.
No problem. Do we still need seats? I see people coming in the back. I see there's a seat right here. There's 2 seats right here. Okay. Alrighty. Dr. Nilanjana Sadhu, is she here? Perfect. Okay. Thank you so much.
Hi everyone, good afternoon, and thank you for the opportunity to share some of the findings from this study. It's a metabolome-wide study of atherosclerosis in an Asian population cohort, which is the Helios SG100K cohort, a multi-ethnic Asian cohort based in Singapore. So there are primarily 2 things that motivated us to do this study. First is that atherosclerosis, it's an inflammatory vascular disease, and the progression of this disease is until a cardiac event occurs, it's silent or subclinical. And about 20 to 30% of adults globally are estimated to have subclinical atherosclerosis. Yet there aren't great biomarkers for detecting early atherosclerosis. So being able to identify such a biomarker would greatly enable effective preventative treatment. The other factor here was that cardiovascular disease is on the rise in Asia as compared to its more stable pattern right now in Europe or in North America. Additionally, Asians are at a greater risk of cardiovascular disease and cardiovascular disease-related mortality. Yet, there isn't as much literature that have focused on Asian populations, and we need to study Asian populations in order to understand what underlies these divergent outcomes. So these are the reasons why we decided to look at the Asian metabolome in the context of atherosclerosis. But why metabolomics? It's just that everyone has their own favorite. We decided to look at the metabolome. Metabolite is really close to the disease phenotype, and it can often serve as a good endpoint, disease endpoint biomarker. Additionally, understanding dysregulated metabolic pathways can enable drug discovery. So with that in mind, we performed metabolite quantification using mass spec, the Metabolome's global discovery panel. So we used that panel to do metabolite quantification in 10,000 individuals in the Helios SG100K cohort. And after quality control, we ended up with a study cohort of 8,124 individuals with metabolite data on 883 metabolites. And this graph over here shows the distribution of the metabolites in different categories. So majority of these are lipids, followed by amino acids, and then xenobiotics. Now coming to the demographic of the study cohort, the phenotype data, I would like to acknowledge Theresia Mina, who carefully and painfully curated the phenotype data. The cohort consisted of adults whose age range from 0 to 85 years, and 59% are females. We have 3 major ethnic groups. We have 67% Chinese, 19% Indians, and 14% Malays. You can also see the lipid profile of this population, the study cohort on the slide. The LDL is slightly on the higher side. And also you can see the prevalence rates of some of the common cardiovascular risk factors, which are very comparable to Singapore's national prevalence rates. Finally, we have carotid intima media thickness, which I've highlighted on the top right. This is a well-known marker of subclinical atherosclerosis, and so this was measured using 2D and 3D ultrasound imaging. This work was led by Rinku Dalan, and if you look at the distribution of this phenotype, you'll find that the mean is around 0.6, which is comparable to the global average of mean IMT. There is also a right tail in the distribution, which reflects the aging population and the higher risk of atherosclerosis in this aging population. So now coming to the analysis of this study. We performed metabolome-wide association analysis for all of these 83 metabolites with carotid intima media thickness. And we identified 126 metabolites that's associated with this marker, CIMT, independent of age, sex, ethnicity, and cardiovascular risk factors such as BMI, blood pressure, cholesterol, type 2 diabetes, and smoking. Now then, for these 126 metabolites, we also conducted genome-wide association studies with genetic data that was available in this cohort. And the summary data from the genome-wide association studies was then used to perform downstream analysis, such as Mendelian randomization studies and co-localization studies that would help us understand or identify potential causal mechanisms. Thankfully, Konstanze has already talked about Mendelian randomization and co-localization, so I will skip over that. So we did an MR scan of all these 126 metabolites that were identified in the previous step. And here metabolite was the exposure, so we wanted to understand the effect of metabolite on coronary artery disease, which is the outcome. The summary data for the metabolite was from our own cohort, which is Helios, SG100K. And for the outcome, we took the summary data from Biobank Japan, which is actually one of the only Asian cohorts with CAD GWAS available. We found that there were 2 metabolites that showed initial evidence, and then only one of them survived further robustness and sensitivity tests. And this is a lipid metabolite called 3-beta hydroxy 5-cholestanoate. So as you can see in that pathway over there, 3BH5C is a metabolite of cholesterol in the acidic pathway, and it leads to primary bile acid synthesis. So what did we find? We found that 3BH5C has a protective effect on CAD, which means that higher levels of this metabolite associates with lower risk of coronary artery disease. We also found that this effect was much stronger, 5 to 6-fold stronger in Asians than in Europeans. I haven't shown the Manhattan plot here, but the Manhattan plot, the GWAS Manhattan plot of this metabolite shows a strong signal in a single locus on the chromosome 11. So we fine-mapped that region and identified a candidate variant. And this candidate variant associates with a number of different cardiovascular outcomes in other population cohorts. Interestingly, it does not really associate with anything other than cardiovascular outcomes, which was kind of a relief. We also found that there is strong global variation in the allele frequencies of this candidate variant across different populations, and that it is much more common in Asians and South Americans as compared to Europeans and Africans. So then we wanted to understand, so sure, the metabolite is associated with coronary artery disease. What gene is manipulating the levels of this metabolite? So we performed co-localization studies using gene expression data from our own cohort, Helios, from GTEx, and protein expression data from UK Biobank. And we identified that a gene called ferredoxin-1 was modulating the levels of 3BH5C. So they shared causal variants. Now going back to that cholesterol metabolism pathway, it's CYP27A1 that metabolizes cholesterol to 3BH5C. And FDX1 interacts with CYP27A1. So much so that it's critical for its enzymatic activity, and pathogenic mutations that alter the interaction between these two lead to a Mendelian disorder, which also shows premature atherosclerosis and neurological phenotypes. So we had this really interesting statistical finding, and we wanted to follow it up with some functional validation studies. So for this, we collaborated with researchers from NUS and NTU. So first, there are two things that we wanted to check initially. The first thing that we checked for was whether changing FDX1 expression, so knocking it out or overexpressing it, was also changing the 3BH5C synthesis. This was done in human and murine hepatocytes and macrophages, as well as human embryonic stem cell derived macrophages and hepatobiliary organoids. And this work was led by Roger Fu, Hao Jie, Mick, Chester Drum, and Leroy. The second set of experiments that we did was to understand the effect of modulating FDX1 on cholesterol efflux. And this work was done by Christine Chung and Kai Yi. So here is a summary of our findings. We find that depleting FDX1 leads to reduction in the 3BH5C levels in the hepatocytes and macrophages, and that treating it with excess of the substrate, which is cholesterol, does not change this fact. Also, overexpression leads to increased synthesis of 3BH5C. We replicated these findings in the human embryonic stem cell derived cells and the hepatobiliary organoids. We also found that if FDX1 expression was rescued, it also restored 3BH5C synthesis. So these set of experiments tell us that FDX1 is critical for 3BH5C production in that acidic pathway of cholesterol metabolism. Our final set of experiments, which was to look at cholesterol efflux, we found that depleting FDX1 also attenuated cholesterol efflux in human aortic smooth muscle cells. So to summarize, we performed a metabolome-wide study of subclinical atherosclerosis in a deeply phenotyped multi-ethnic Asian population cohort, and we identified 3BH5C, which is a lipid metabolite, as an independent biomarker of atherosclerosis. We also found that it is strongly associated with cardiovascular disease in Asians, and that the effect is far stronger in Asians than in Europeans. We also identified ferredoxin-1, the gene, as a key regulator of 3BH5C synthesis, and we found that altering FDX1 also alters cholesterol efflux. So this paper is currently under review. It's on medRxiv for anyone who wants to read it and give us some feedback. And with that, I would like to thank Prof. John Chambers, who has guided me on this study. It's a cross-functional, cross-institutional study, so there are a lot of people to thank, some of whom are listed on the slide. I would like to thank the participants of our Helios SG100K cohort, and also the operational and administrative staff who actually enable the study and collect the data. Thank you for being such a great audience, and I would love to take questions. Okay. Thank you.
Okay. We have one more presenter. There we go. Do you want to plug in your laptop? No? Okay. Let me see. Very good. Okay.
sorry for the technical difficulties. So I'm a student from NUS working in Chester Drum Lab, and I'm talking about heart failure today. And then, okay. If we classify heart failure according to ejection fraction, there are reduced ejection fraction and then preserved ejection fraction. And then there are quite a lot of treatment established for reduced ejection fraction treatments. But then there is not that much treatment for preserved ejection fraction. So our group has initiated a metabolomic study of 2 existing heart failure cohorts. And we try to find if there are any insights we can draw from the analysis. 2 aims of our study. The first aim is we want to see if there are any metabolomic difference between heart failure with preserved and reduced ejection fraction. And the second is we try to associate metabolic patterns with 2 years outcomes. The cohort we are using in this study is the people and shop cohort. They are 2 sister cohorts initiated by Prof. Mark Richard and Rob Doherty in New Zealand, and then also Caroline in Singapore. And then the main study was performed and concluded in 2018. And then now we are reusing the plasma biobank of the samples. And then we try to analyze the metabolomic differences between the plasma samples. All the heart failure patients are prospectively recruited 6 months after the first episode of heart failure decompensation. And then we monitor the 2-year follow-up according to, in terms of the all-cause mortality and the heart failure readmission. There are 3 phases that I will present today. There are more analysis we have performed, and yeah, because of time limit, we won't present all of those. The first phase is we try to identify the metabolite enrichment in heart failure that is common to both the Singapore and the New Zealand cohort. And then we try to, in the second phase, we try to associate this metabolic signature to the outcome in HFpEF and HFrEF distinctly. And then in the third phase, we try to see what is the cause that is contributing to the difference in terms of the association between the outcome and then the metabolic composition. First result, you can see from the left is a very standard tally. We used the weighted correlation network to construct metabolic co-expression network for the New Zealand cohort of people. And then this is constructed across all the heart failure patients. And then on the right is the metabolic network constructed for the Singapore cohort, again for all the heart failure patients. There are 760 patients for each of these cohorts. So after we identify all the modules and then label them with their functional involvement according to all those functional databases like CAT, Reactome, Wikipathway, etc. We find 4 cohorts, 4 modules in common across the 2 populations. They correspond to the endothelial regulation, the arginine NO, arginine nitric oxide regulation, and then inflammation. Also, there are 2 other long glucose energy flexibility related modules, which are the free amino acids, and mainly is the branch-chain acyl-carnitine and essential amino acid. Yeah. And then the second result we find is that actually all of these 4 common modules are associated with 2 years outcome. But then they associate with the outcomes in preserved ejection fraction and reduced ejection fraction differently. So you can see at the top, you have a look at the forest plot. You can see it's the top modules and the bottom modules that are most associated with HFpEF mortality. And then these modules are actually corresponding to the endothelial regulation. And then for heart failure with reduced ejection fraction is the middle two modules that are more associated with HFrEF outcome. And then this corresponds to non-glucose energy metabolites. And then when we try to visualize how the quintiles, so it's like the every 20% increase or decrease in these metabolic expressions associated with the outcomes, we see that the trend remains consistent for both the New Zealand and the Singapore population. And then, as all of us may know, the nitric oxide pathway is protective against 2 years mortality. But then is the inflammation pathway is harmful for 2 years mortality. And then, yeah. So this result is quite consistent with our understanding. So the third aim is to see whether we can explain why there is such a difference in terms of the module outcome relationship difference in HFpEF and HFrEF. First, we compare the connectivity patterns of the different modules, and then actually they are quite similar in terms of within HFpEF and within HFrEF. But then what the difference lies in is the module composition. When we focus on these two networks, they belong to the arginine nitric oxide metabolism. You see, there are a few essential amino acids and then glucose regulation metabolite. They are only incorporated into the network in HFpEF, but not in HFrEF. And when we also try to see the other metabolite and label their functional involvement, we find out a few other metabolites referring that they are involved in oxidation or nitrosative stress. They are also incorporated in the network, as well as the already established arginine nitric oxide metabolite. So this is quite interesting because this observation we have is in accordance, in alignment with a previous publication that states that actually the cause of heart failure with preserved ejection fraction is due to meta-inflammation. And then in specific, they talk about the upregulation of microvascular dysfunction, increased nitrosative stress, and increased insulin resistance. And similarly, what we see is that we see the involvement of nitric oxide metabolism, and then the involvement of free radical regulating metabolite, and then some glucose regulating metabolite in our network. And then this network is only enriched in HFpEF, but not in HFrEF, and then it is also associated with the outcome in HFrEF only. So yes, so that's the end of my presentation today. Yeah, thank you.